Okay, well, thank you very much for coming. Uh, welcome. We are SynBio.Oxford, a student-run society based at the University of Oxford to promote synthetic biology. And we organize these talks with experts in academia and in industry. And well, we've switched to delivering them as webinars, but we're quite happy that we can share them now a bit more globally. And in order to do this, we get some support. So a big thank you to our sponsor, Integrated DNA Technologies, our IDT, that help us continue these webinars. So the format of this will be quite simple. After this quick introduction, Lucia will give her talk, and that will be followed by a question and answer session. And you can put your questions into the chat box, or you will then be able to raise your hand and we'll sort out the order. So for today's talk, we've invited Dr. Lucia Marucci from the University of Bristol. She got her bachelor's and master's degree in mathematics and informatics. And from that, she followed on with a PhD in automation engineering at the University of Naples, Federico II. And during that, she worked already on mathematical modeling and analysis of synthetic gene regulatory networks. After that, she was a postdoc, an EMBO fellow at the Center for Genomic Regulation in Barcelona. And since 2013, she's at the University of Bristol, where she's now an EPSRC Early Career Fellow and Associate Professor for Systems and Synthetic Biology. So thank you very much for coming today, Lucia.
Thank you so much, Corinna. Thanks for the invitation. It's really my pleasure to be here today, being able, even if not in person, to talk to you about research that is going on in my group. Let me share my presentation, which I hope you should be able to see. Yep.
Yes, all good.
Great. So, as Corinna said, I'm based in Bristol, and I am leading an interdisciplinary group that is particularly interested in modeling and controlling dynamics in mammalian cells. Why are we interested in this kind of mainly nonlinear dynamics is because it has been well reported that complex dynamics in space and time of gene expression can drive specific cell fates and phenotypes both in cells and in organisms. In these slides, I'm just reporting a few examples. It is well known that oscillations in space and times of genes of the Wnt pathway, such as AXIN2, tightly control somatogenesis during development. We and others have reported the importance of both doses and dynamics of various genes in the control of pluripotency of stem cells and in their differentiation. I will talk about this in a few minutes. And also in other processes, dynamic matters. Here is an example of a cancer cell, and this is the RAS-ERK signaling pathway. It has been reported that providing different kind of dynamical input upstream of the pathway, as you can see here, the downstream response, looking at ERK, so a downstream gene, is different if considering normal versus cancer cells. So this is another example of the importance of temporal dynamics to determine the phenotype of a cell. And for sure, if moving to cells, we look at tissue, there are also different kinds of dynamics that matter. For example, mechanical forces among cells and the tissue environment. So the goal of my group is basically to try to answer these questions. We are trying to combine maths, computational tools, and experiment. Firstly, to try to understand how this kind of collective behavior is achieved. What is the role of expression dynamics for cellular decision making? And also, we are trying to combine tools from synthetic biology and control engineering to directly control gene expression to then affect cellular fate. I will now start with a few slides showing you some mainly systems bio results in which we try to basically answer these first two questions. And I will then move to the core of the talk that it will be instead focused on the control of this kind of dynamics. Let me briefly introduce for the mammalian cell work the key tools that we use in the lab. To study pluripotency and differentiation, we mainly use mouse embryonic stem cells. As in these cartoons, mouse embryonic stem cells can be isolated from the inner cell mass of mouse. And they can be isolated during different stages of development. For sure, if you isolate cells when you are at this or this stage, so in a further stage of development, the cells will be more differentiated as compared to cells isolated earlier. We work with cells isolated here, so they are really pluripotent cells, but actually not just the isolation stage, but also the way in which you culture your cells in a dish matters. I will show you during the talk various results with cells cultured either in this medium that is called serum leaf. This is the most traditional medium to culture cells, and it has been well reported to keep cells in the between of a naive ground pluripotency state and a more primed for differentiation stage. And I will also show you some results instead in 12-leaf, which is a media which does not contain serum, it's serum-free. It contains 2 inhibitors. I will show you later results in which we play with these 2 drugs as control inputs. And this media should keep cells in a more naive ground state of pluripotency. Okay, so starting with these results, quite old at the moment, I would say, more from the systems bio kind of approach. What we wanted to do was to try to understand how dynamics of specific genes were possibly regulating pluripotency. We started focusing on beta-catenin, that is the main protein of the Wnt beta-catenin pathway. It's a pathway which is important also for other processes such as cancer. And to track the dynamics of this gene, we tagged beta-catenin with a GFP. At the time we used zinc finger, but anyway, we inserted this GFP on one allele in the genome of a cell line of mouse embryonic stem cells. And then we started to look at this GFP as a proxy for beta-catenin expression. So here are some immunofluorescences. This gene here is called NANOC. This is a mast cell regulator of pluripotency, which was already well known to be heterogeneous when cells were cultured in serum leaf. We actually also noticed that also beta-catenin was heterogeneous, not just when looking at a few cells by mono, but also when analyzing a higher number of cells by FACS. You can see that as compared to negative control, we have in our tagged cell line the coexistence of a population with low beta-catenin and high beta-catenin, again using GFP as a proxy for beta-catenin expression. So we observed this heterogeneity in the expression of beta-catenin, and actually I'm not showing you, but if you're interested, all the data are in this paper. We also noticed the fact that cells were able to switch from a state with low beta-catenin to high beta-catenin and vice versa, so also fluctuations in times. We then asked, okay, why is this happening? And to do that, we developed some models. We basically described with a quite standard systems bio approach, the dynamics of this gene regulatory network that we have got here. Without going too much into the details of the bio, basically the majority of these interactions were known, but this inhibition here that we proved experimentally with a range of different sets of experiments. And we developed a differential equation model describing the dynamics of these 4 genes, basically. The model is quite standard. We used Hill kinetics to describe both activations and inhibitions. We assumed that the dynamics of the mRNAs were much faster than the protein dynamics, so steady state approximations for mRNA, and also the model is stochastic. So we have these Gaussian terms here, which are basically describing the effect of noise. Nice. So as with all models, usually firstly you feed it to experimental data. We use some parameters found in the literature. Other ones were fit on experimental data we generated. Indeed, for example, we measured again by flow the steady state distribution of both beta-catenin and NANOC. And the model was able quite well to recapitulate the stability observed by FACS. And after we had a model which was fitted, we then asked to the model, why are we having the stability? So where is this heterogeneity coming from? And to do that, we performed a numerical continuation analysis, basically trying to understand the bifurcation behind this kind of dynamics. And what we found by continuing the steady state of our 2 genes of interest, again, NANOC and beta-catenin, against one of the parameters of the system, is the fact that, as you can see, we basically have, for both genes, the coexistence of 2 stable steady states are the solid lines. separated by unstable solutions, these gray lines. So basically what the modeling is telling us is that it looks like in serum leaf, when we look at cells, we have cells with low and high NaNOC and beta-catenin because the system is bistable. So the model is basically giving us hints into the dynamic properties which cause this phenotype when we look at our cells. We also tried to look a bit at the bigger picture, both in terms of other genes, because not just beta-catenin and NaNOC, but also other genes are important to maintain pluripotency. And secondly, because also we wanted to capture dynamics in 12-leaf. So as I mentioned earlier, 12-leaf is this media which is serum-free, which contains 2 inhibitors, and is supposed to keep cells in the ground state of pluripotency. So the expectation would be that when you culture cells in this media and you look at NaNOC, that is a marker of pluripotency, all your cells should be switched on for NaNOC, okay? Actually, that's not the case. So these are data from this paper here. They generated a reporter cell line for NaNOC. And again, looking at GFP, that is tagging NaNOC, they found that in serum leaf, again, we have got traceability, fine, but also in 12-leaf, we still have a bunch of cells that are low for NaNOC expression, so that are basically not in the ground state of pluripotency. This is steady state data, and when performing time lapses to basically see the transition of cells from low to high NaNOC, they found that also cells in 12-leaf were fluctuating. We wanted again to understand this with the help of maths. So what Simon did, a former PhD student in my group, was firstly to develop a gene network for pluripotency. Without going too much into details, he basically used different data sets published in the literature, and he came out with this gene network which contains transcriptional interactions, are the solid lines, which also contains protein-protein interactions, are these dotted lines, and that importantly contains PD and chiron, which are the 2 inhibitors present in the 12-leaf. So basically, this network should be able to recapitulate what happens in both the media conditions I spoke about, serum leaf and 12-leaf. Again, he developed a model. It's just a bit more complex than the other one, but the structure is similar. So again, steady state approximation for mRNAs, Hill kinetics, and Gaussian noise. And also, as we wanted not just to basically describe the intracellular dynamics, so the dynamics of this gene network, but also the extracellular dynamics, what Simon did was to couple the dynamics of the gene network to a cell cycle model, trying basically to describe how the expression of specific genes possibly affects the dynamics of proliferation of our cells. And this in turn was linked to a cell mechanical model. To do this, we used CHASTE, which actually has been developed in Oxford. It's a very nice tool to perform agent-based simulations for mammalian cells. And these are the results he got. So he was able, with this agent-based version of the model, to recapitulate, again, the stability of NaNOC in serum leaf. These are simulations. This is, again, the experimental data set. But also the fact that in 12-leaf, we still have a bunch of cells with low NaNOC. This is fitting data, validation data. So I used a different data set, basically sorting experiments in which cells were sorted for high or low NaNOC and then repeated. This is experimental data set, recapitulated quite well from the model. And then again, we use bifurcation analysis to try to understand why we basically still have this population with low NaNOC in 12-leaf. And what the fitted model told us is that, again, in serum leaf, we think to be in a bistable condition. So again, low and high NaNOC steady states separated by an unstable solution by 2 saddle node bifurcations. In 12-leaf, it is true that we are here. So we are shifting towards the NaNOC only solution, so which cells should all express NaNOC. But still, we believe that we are very close to the bifurcation point. Meaning that if there is enough noise in our cells, some of them could escape the NaNOC and be trapped into the NaNOC low solution. Okay, so these first few slides were again about some recent systems by results in the group. We believe that these modeling approaches were useful because they enabled us to explain why we were observing this kind of multi-stability and fluctuations. Although still, these results are not telling us why the cells possibly need this kind of stability. So is this stability functional for pluripotency and differentiation? We don't know this from these results. And also I showed you very complex models, especially the last one. It contains lots of parameters. So it is true that we did our best to get some from the literature and to measure them, the one that we could at least. But it is also true that the structure of the model might not be unique. So as every time you develop a model, you do some assumptions, and possibly with different assumptions, you would have a different model that might still fit your data. So what we are doing at the moment is to use a different approach to answer these questions, which is based on synthetic biology. So what I will show you in the next slides are various kind of control approaches that we are using to try to directly engineer different dynamics of gene expression to in turn both understand how they affect cell behavior and then to indirectly try to directly control cell fate. I don't know how many of you are familiar with this field of synthetic bio that is called cybergenetics. And basically it refers to various ways to try to control living cells using principles and tools from control engineering. This is a very nice review by Domitilla Del Vecchio and colleagues, in which a range of different approaches are proposed. These are the two main ones I'm going to talk about today. And at the very end of the talk, I will mention briefly a third approach, multicellular control, we are working on in Bristol. Embedded feedback control, as you can see here, it relies on feedback. So you see that this loop is closed and is based on the fact that both the controlled process and the controller are implemented into the cells via biomolecules. Instead, in silico external feedback control is implemented by basically placing the controller outside. So you see that here our controller is implemented on a computer. The process is still within a cell or a population of cells, so it's still implemented through biomolecules. And similarly, what you do basically is to try to control the Y, the output of your process, but in this case, through a computer. Let's start firstly to show you some tools we have recently developed to enable this kind of controls. This is a tool we published last year, and basically it is an extended TET-on system. So if you are familiar with TET-on, TET-off systems, they're basically engineered construct which enable with drug, usually doxycycline or tetracycline, to tune on or off the transcription of a gene. These tools have been used for many years now, both in vitro and in vivo. For synthetic bio applications, sometimes they have issues, mainly because the dynamics are not very fast. In particular, for the TET-on systems, it has been well reported that the switch off dynamics, so when you remove doxycycline, are slow, and vice versa for the TET-off. To sort this out, we developed this tool in which basically not just we have a TET-on promoter, which again enables to switch on transcription by adding doxycycline, but also what we did was to fuse the gene that is regulated with a destabilizing domain. These destabilizing domains, what they do basically is to degrade your gene of interest unless you add a drug, in this case, TMP. So with this system, as you can appreciate here, what basically we can do is to tune it with two different drugs, one doxycycline that is working at the transcriptional level, and the second, TMP, that is working at the post-translational level. And we proved with different experiments that the system is quite robust. So for example, when the construct is regulating an M-cherry fluorescence reporter, if we look by qPCR at the mRNA of our M-cherry, as you can guess, this is the no-inducted M-cherry, and its level don't change if we add just doxy or both doxy and TMP, which makes sense because this drug is supposed to act just on the protein. While if we look at the protein by flow, you see that we can have three nice peaks. So the one in the middle is the one in which we just have doxy. So basically we are stabilizing the mRNA, but not the protein. And the one that is the most red one, basically this green line here, is the sample in which we have got both doxy and TMP. And then again, we wanted to compare this inducible system with a standard tecton system. We did that again with both models and experiments. This is basically a standard tecton. So the tecton promoter is driving M-cherry, while here the M-cherry is also fused to a destabilizing domain. We developed two ordinary differential equation model, in this case no noise, to compare the two systems. They're quite similar, but here I just have doxycycline regulating the transcription of my M-cherry. There is, as usual, any function because we found that there is saturation. Here, I also am describing with this term the fact that with TMP, I can block the destabilizing domain induced degradation of M-cherry. Again, with any function because we found that there is saturation also for TMP. And we found that the system works quite well. So when looking at steady state levels of our M-cherry in the full system, in which also we have the DD, you see that these are model simulations. These are comparison of model and data. We have a very good range, so we can really tune very well the levels of our M-cherry by playing with both concentration of PMP and doxycycline, as we saw by simulating the model and comparing the model with experiments. And importantly, we found out that our system with the destabilizing domain switches off much faster than the standard tecton. So what we did here was firstly to induce the cells, here just with doxy, here with both doxy and TMP, and then to remove doxy or both doxy and TMP. And I hope you can appreciate these dotted line are model predictions, while these ones are experimental data, that our cells switch off quicker. So it looks like this system can be very good for tuning, not just the steady state levels, but also the dynamics of genes of interest. And we started to do that. For example, again, beta-catenin. I told you earlier that we found that the cells have heterogeneous levels of beta-catenin, our mouse embryonic stem cells, but we couldn't understand why, if that was something useful for cells. And with this tool, we are now starting to test that. So what we did was, first of all, by CRISPR-Cas9 to generate a new mouse embryonic stem cell line in which we completely deleted beta-catenin by CRISPR. We did that in such a way that we could have a system that was very clean. So there was no interference of endogenous regulation. Indeed, in this beta-catenin knockout cell line, we then stably inserted our inducible construct in such a way that then with the drugs, we could just play with the exogenous beta-catenin. And then we started to look at phenotypes of pluripotency with different experiments. This is just an example. This is called an epistaining. Basically, it's a staining that allows you to count the pluripotent colonies that you have in a dish. So basically, the more stained colonies you have, the more pluripotent colonies you have. And what actually we found is that in specific media culture conditions, basically these two and a bit also this one here, there is actually an effect of the doses of our exogenous beta-catenin on pluripotency. Because in this case, we are not perturbing the dynamics, just the steady state level. So the doses, you can see that with lower levels of doxy here, we have gotten higher number of stained colonies as compared to this condition in which doxy is higher. So this tool enables us to play with doses and check the effect on pluripotency. What about dynamics? So this tool so far that I showed you can be used mainly in open loop. So we give doxy and TMP, and then we check what happens to the exogenous M-cherry or M-cherry fused with beta-catenin, as in the last slide. And then we try to match those doses to pluripotency phenotypes. Playing with dynamics can be a bit more tricky, and this is where closing the loop can really be beneficial. And to do that, we are mainly relying on external in silico feedback control. So this is the cartoon I showed you earlier. The controller is implemented outside. And down here, we have a standard closed approach in engineering. So we have got a controller that through an actuator can modify our system of interest, okay? And we want to measure specific outputs of our systems. We need some sensor there. And in turn, to compare these outputs with the reference. The reference will basically be our target, our desired output. So by comparing the reference with the actual output, we will have an error unless they're exactly the same. And the controller, in turn, by closing the loop, will try to minimize this error by acting on the system. This is a very general principle in engineering. And this is our implementation for biological systems. We are using this for mammalian cells, but actually also for bacteria. I will show you at the very end some results with bacterial cells. So the control algorithm is implemented externally by a computer. What this computer does is basically to compare the actual output of cells with the reference and to act on the cells by changing the media that is provided to cells. To do that, we rely on microfluidics, mainly because we need to be able to dynamically change the media to cells. So that's why we are using microfluidics. And to sense, what the cells do, we use time-lapse imaging. So we basically continuously image the cells. We measure the outputs to date mainly using fluorescence reporter. And every time we take an image, we process the image. So we use online segmentation algorithms. These segmentation algorithms, by quantifying the fluorescence of protein, allow us basically to compute the output, which again, in turn, is compared to the reference. Error is computed, and so on and so forth, and we close the loop. So in these slides, I have some further details about our platform in Bristol. This is the device we used so far. It's a device that was developed by the lab of Jeff Hasty in California. It's a PDMS1. We have got multiple chambers here, and importantly, we can mix on-chip two media. These two media will be basically, as in the examples I will show you, either plain media or media with the control inputs. The chip is secured on the stage of a microscope, as you can see here, as we do the time lapse. And in turn, this is connected by fluidic lines to our syringes. So these syringes basically are our actuators. And what we do when we perform a time lapse is to automatically move these syringes in such a way that the input that is provided to cells changes in the way the control algorithm tells us. So let me show a typical experiment. This will be more clear, I think. So again, these are mouse embryonic stem cells, which carry the construct where the cherry is regulated by the doxycycline with the teton and TMP for protein stability, okay? And in this time lapse, we are basically trying to steer our cells to track this signal. The control reference is this blue line. The control algorithm we used here is a very simple one. It's called the relay control algorithm. It simply provides the inputs if we are below the reference, while it removes the inputs if we are above. So it's a very, very simple one. It's completely model-free. But still, as I hope you can see, it's able to steer the cells to track this time-varying signal. On the right is a similar experiment. So again, is a very basic relay control algorithm. There are two differences. One is that here we are controlling beta-catenin. So the mCher is fused to the beta-catenin gene in our constraint. And here the reference is just basically reaching a steady state value, 50% of the maximum fluorescence. We are also trying to develop more sophisticated control algorithms, such as model predictive control, which are have been well reported to be much more robust to noise. Briefly, this kind of algorithms instead need a mathematical model of the system. And what basically they do, if for example, we want to track this red line here as a reference trajectory, what they do at the time, let's say here, in which they need to decide which media to give to cell, is not just to use the actual output of the cells, but also to use a mathematical model of the system, okay? So as you can see here, when our controller acts, it uses the model, it uses the actual measures, and then it basically tries to optimize a cost function in such a way that it tries to reduce the error in the next step. It's more powerful because it also uses a mathematical model of the system. That's the key idea. And it predicts the inputs to provide over some time in the future, and that is called the prediction horizon. We have used different kind of model predictive control strategies so far, not adaptive ones yet. This is something we're trying to do at the moment. With adaptive strategies, what you do that actually is better is to fit your model as you do your experiment. It's better because of noise. So maybe not each experiment is equal to the others. We didn't do that yet, but what we did was firstly to perform some open loop experiments where basically we were providing pulses of the drugs to cells, we were measuring the outputs, and we were fitting a quite simple linear model. This model was then used by model predictive control. And as you can see here, model predictive control worked quite well, again with the same cells, with the same construct, to steer them to track the signal. Not showing you in this talk, but if you're interested, have a look in this paper. We also managed to perform single cell control. Here we didn't because these cells grow in very big colonies, but we can also control just one cell over the time lapse if they're flat enough. So if the segmentation works to segment and track just one cell and its progeny over time. Okay, so we have this platform. It works quite well. How are we going to use it? Going back to the first part of my talk, I was showing you those models that were basically telling us, for example, thinking again about nanog. Nanog is bistable, that's why some cells have it on and some cells have it off. To find that out, we needed to do experiments. We developed models and then we extracted features from the model. This is basically the typical thing you do, not just in biology, but in various applications. What we are trying to do at the moment is basically to do the same, but without the need of models. Why? Again, to avoid issues in case we don't trust too much our model. And to do that, we are trying to apply something that is called control-based continuation. Here are a few key papers that talk about this. It's a methodology which so far has been used quite a lot for mechanical systems, but never for biological systems. So the idea is again to basically use our platform to close the loop, but to use control strategies which don't need the model, and then to try to extract nonlinear features of our system directly from experimental data. These are unpublished results. We didn't yet perform the experiments, but instead what we are trying now is first of all to prove in silico with models that this is feasible. For example, again, nanog. I told you that is bistable, and we saw that by FACS and by mono. If I would perform an experiment in open loop, in which I change whichever bifurcation parameter of the system, what I would observe are just the low and high steady states. Why? Because they're stable. So unstable solution, I will not converge there. I can't observe those solutions. However, the idea of CBC is to basically design a smart control reference, for example, a step function, in such a way that if I can force the steady state of nanog to track all these solutions, including the unstable one, I should then be able offline to hopefully reconstruct the entire continuation curve, in which not just I've got my stable solution, but also the unstable branch. This is a simulation, so here we use the model to generate in silico data, but it seems that it's feasible to do that as far as we can keep the controller non-invasive. And in this example, the controller is a very simple one. It's simply a proportional controller. We didn't do experiments yet, but what we are starting to do now in the lab is anyway to check if we can control endogenous genes of pluripotency in mouse embryonic stem cells with simple algorithms. And it seems to be the case. So here are just a couple of time lapses in which, again, with a simple relay algorithm, we are trying to control, in this case, an endogenous gene, REX1, that is a target of nanog in mouse embryonic stem cells, with these two drugs, that are the drugs that are present in the dry leaf media. So it seems that we should be able to do CBC experimentally and to reconstruct the full nanog S curve. And also, we would like to use CBC for a different kind of application, mainly to prototype synthetic networks. Why? Because also with gene regulatory networks in synthetic bio, usually what you do is to design your system, you develop a model, and then you check if your model, for example, is bistable. Here is an example. This is the very recently proposed toggle switch, Nikolai from Lugagne and colleague. Is a toggle switch supposed to be bistable. So in the paper, they developed both a deterministic and a stochastic model, and they found that, for example, plotting the north lines of the steady states of Tetar and Lakai, the two genes, there are three steady states: usual thing, two stable, and one unstable. Question is, can we use CBC to prototype network like this? Again, we didn't do experiments yet, but we are performing simulations at the model. Irene, a PhD student in my group, is working on this. It seems that it's the case. So also here, she's using a proportional controller. In these simulations, she used the ODA model, so the deterministic model for the toggle switch. And here, the dotted line is the continuation curve from the deterministic model, while these points here are the CBC, the control-based continuation points. It seems that if, again, we design smartly our reference signal, we can force the steady state of theta, in this case, to basically go through all the solutions, also the unstable one, as varying IPTG, one of the two inputs. And it seems to work quite well also if the system is noisy. So here she did the same, but she used the stochastic version of this model. Actually, this is a fully stochastic version, so it's a Gillespie simulation, and still, although not perfectly, our CBC point more or less lie on the actual bifurcation curve. Next step is to do experiments, for sure, regarding this. Second application of the platform. This is something that we started to do quite recently within the aims of my fellowship. We want to exploit CBC to optimize culture condition for stem cells. Why? Because it has been reported that keeping cells for too long into a leaf, so again with these two inhibitors, you can start having issues with the chromosomal stability of your cells. So the question is, can we keep cells pluripotent, maybe just giving pulses of these drugs instead of keeping that all the time? And that's where we think that control can help in telling us the optimal combination of drugs which keeps pluripotency without basically damaging cells. And we're planning to do this across scales, not just in microfluidics, but also working with bioreactors. And also the idea is to use, again, modeling, agent-based models to compare the response of cells across scales, as we expect that response to be different. We also are planning to start using human cells, which we think is very relevant. And finally, something which, again, we started quite recently, is to try to use the platform to design in a smart way combination therapy for cancer. Mainly, we are focusing on a specific type of lung cancer for which it has been reported that continuous exposure to drugs can cause resistance. So again, we would like to find an optimal combination and timing of exposure to drugs that minimizes this. So the platform is the same, but we are going to use chemotherapeutic agents as drug, cancer cells as our plant, so the cells that we want to control. And in terms of control outputs, we are going to look at pathways involved in cancer. And the idea is similar. So starting in open loop, giving specific patterns of drugs, measuring the outputs and using these input outputs data to generate black or gray box models. And then to close the loop. So for example, to set the control reference as a specific, for example, profile of proliferation or activation of specific signaling pathway outputs, and then exploit the control action to understand the optimal combination of drugs. We are not there yet because we started very recently, but what we are starting to do at the minute is to basically culture non-small cell lung cancer cells, PC9, in the device. We are generating cell lines in which we are tagging these genes with a star that are key genes in the pathways involved in cancer, while looking for drugs that tax at different levels of these pathways. And Adil, a new postdoc in the group, is also trying to design and construct new microfluidic devices, which first of all allow us to play with more than 2 drugs. That is what we can do with the current device. So here you see we have got 4 inlets. This is just a console simulation in which is basically changing the media into this inlet to check that the mixing is working. This device also is easier to build because it's just a layer of PDMS, and importantly, the chambers are bigger. So we are now starting to see if we can load human cells here and human embryonic stem cells here, and it seems to be the case. In the other one, they didn't fit. And finally, regarding this, in collaboration with Tom Gorochowski in Bristol, we are also developing more robust segmentation tools. Why? Because we do the segmentation online. So as you can guess, if the segmentation goes wrong, all the control experiment will be wrong. And we actually found, as in this example, that sometimes our current algorithm, which is a simple one based on Otsu, fails. For example, here, you might be able to see that we have got the chambers of our chip. The Otsu sometimes can't discriminate between the edges of the chamber and the actual cells. So if that happens, it will be a problem, because if, for example, we are computing the average fluorescence of cells, the calculation of the area will be wrong. So the entire calculation of fluorescence will be wrong. Instead, we just developed Cheetah, that is a tool in Python, which is based on deep learning. It uses a UNet architecture, which has already been reported to be very robust for segmenting images. And we found Cheetah to be both robust and also nicely, it can discriminate between alive and dead cells in such a way that in the quantification, we can just look at alive cells and get rid of whichever quantification of cells that really we don't want to follow and to control. The last 5 minutes of this talk, I want to just briefly, very briefly introduce you a very different kind of approach that also we are attempting in Bristol, at the moment in bacteria, but with the plan to switch soon also to mammalian cells. This work is in collaboration with Mario Di Bernardo, Nigel Severi, and Claire Gerson in Bristol. So the idea here is to implement the control in a different way, mainly for two reasons. The first thing is that every time you implement a controller, you're going to insert lots of exogenous DNA into your cells, and that can cause burden. And the second issue is modularity. So the more exogenous stuff you insert, the more, if you want to change something, it is a problem because your system might not be very modular. So to sort this out, what we thought is to basically change our approach, our current approach, in which basically the majority of the parts are into one cellular population and the controller is outside, with this approach that we call multicellular control. And the idea is quite simple. So basically we want to divide the various functions of regulator, actuator, process, and sensor into populations, again, with the aims of reducing the metabolic burden and increasing modularity. So if working, we should have something like this, two different strains of bacteria. At the moment, we are working on bacteria. And in principle, if providing a reference to the controllers, the targets should track that reference, as you can see here. We proposed this just in silico so far, although we have got some data for the controllers. And that's our implementation. We are now building all the parts in the lab. Basically, the idea is to have this module in the controllers that enables basically to compare the reference signal with the signal from the targets. In turn, this computed error will generate a signal that we need to transmit to the target, and that is done via inducer molecules. These inducer molecules in the target cells, in turn, will need to inhibit a gene. Why inhibition? Because we need overall a negative feedback loop for basically the control action to work. And finally, to close the loop, we need again to feedback this signal to the controllers. And this is done through another signaling molecule. Right, as mentioned, we don't have yet in bacteria a full biological implementation of these different control strategies, this multicellular control strategy. We at least have simulations. So these are simulations that Antosh, a former postdoc in the group, performed in BSIM, which is a software we developed in Bristol. The nice thing of this video is that anyway, the geometry of the chamber is the same as the device, the PDMS device we are using. This chip is different from the mammalian one, as you can see, but is again a PDMS device. And if everything is working, in principle, again, we should have the targets in red here and here, tracking the reference that instead is provided to the controllers. So we don't have yet the full circuit again, but we have the masters working, the controllers working. We implemented a comparator in an E. coli strain, which is based on a sigma anti-sigma pair. Basically, what happens here is that the formation of the complex is regulated by two molecules. One is a PTG that in the full circuit will be used as control reference. And another one is HL that in the full circuit will be provided by the targets, and now we can provide externally. And now we have a GFP that we are using to basically see if our comparator works. And it seems to be the case. So these are steady state data here, 5-hour response of E. coli strain with this construct. And you see that we have got a very good dynamic range of response to both IPTG and AHL, where again, IPTG is basically inhibiting our GFP because it's promoting the anti-sigma, and vice versa for DHL. And also we are starting now to test, we recently published this paper in which we are testing this strain in microfluidics. That is where we want in the end to test the entire population for multicellular control. To do some tests, we applied again external control here. This is very similar to what I showed you for mammalian cells, again with the relay control algorithm. And you see that we can control the population, in this case using as input just IPTG with AHL fixed. Where is this going next? At the moment, we are building the circuits for the target population with the aims of then testing the full multicellular control, both in microfluidics and in bioreactors. And then the next steps will be to test this for mammalian cells. So both with 2D cultures and then hopefully for multicellular controls within organoids. And with this, I want to conclude my talk. I want for sure to thank all the people in my group that developed this work. This picture is not very updated. As you can guess, this year has been complicated to take picture with the group, but the names are updated. The people highlighted are the ones that generated the results I show you, former and current member of the group, all the collaborators, all the founders, and all of you for your attention. Thanks again. And I'm more than happy to answer any question you might have.
Thank you very much, Lucia. So how we can do this question and answer session is that you can write your questions into the chat box, or you can click the raise your hand. And I've allowed you to unmute yourselves now. So if you want, you can ask the question personally to make this a bit more interactive. So first up, we have Shun Tang. Would you like to unmute yourself and ask your question?
Yeah, for sure. Can you hear me now?
Yes. Yes.
Yeah, thank you very much. That's a very interesting talk. So I think I have two questions. The first one is the one I put in the chat box. So for the control, where you mentioned it's a model-free control, the first part, I think. Mm-hmm. So how do you, I was wondering, how do you tune your controller to decide what actually, like how much dose you're going to inject into your system? And also, I mean, this related question. First one is how do you tune your controller? And then how fast are you able to do the image processing in order to provide a feedback? So I guess the reason why I ask this is, how is your feedback effective, or how is your control action effective to manipulate the system? Yeah.
Thank you so much. So how often we update the control action? In the first experiment I showed you, so I mainly showed the experiments either with the relay controller or with an MPC controller, because the one with the proportional, there were just simulations. For the relay controller, we update the control action every hour. We don't take images more frequently because the time lapses for mammalian cells are quite long. They're usually 2 or 3 days, and we don't want to basically damage the cells with photobleaching. So we take an image every hour. Being the controller model free, in that case, we just act when we take an image. The processing of an image is very quick because in all those experiments, at least we are using an algorithm that is based on Otsu thresholding, is really, really quick. Another thing that is important is the actuation time. Those experiments are performed with syringes. It takes more or less 5 minutes from when we move the syringe for the new media to go into the chip. So that's the only kind of actual delay that we have got. 5 minutes, the time that the entire media in the chamber is changed with the new control input. For the relay controller, going back to your other question, we have nothing to tune for 2 reasons. One is that it's a relay, so we just decide to give the input. If the inputs are switching the system on, we just give it if we are below the reference, or we remove it if we are above. All those experiments that I showed you, they are basically bang-bang controllers. So we don't play with the concentration. Is there or is not there? Anyway, if you are interested more into these, if you look into the Postillon et al. 2018 SCS paper, there we also performed experiments in which we were playing with the concentration. And at least for these systems, it didn't really matter. So the effectiveness of the control action was similar, at least for those, but these are quite simple exogenous systems that we are controlling. Maybe for endogenous ones is different. Something else that I wanted to mention is when we apply MPC, we have got more freedom there, because still we take an image every hour, but our model is continuous, right? So when we apply MPC, we can also, if for example, I take an image now and another in 1 hour, I can still change the media within this interval, because I have the model that can tell me what to do in that hour. And in that case, we change the media not earlier than every 15 minutes. The reason is because we found by trial and error that the cells get a bit stressed if we change the media too frequently. There is a bit of shear stress anyway in the chip. So that's our constraint. And the last thing I want to mention for completeness is that I also showed you experiments in which we use the deep learning algorithm. Now, when using the deep learning algorithm, because we don't want delays, we trained the network before. We couldn't do the training of the actual UNet network online. That would take ages, because it's true. But if we train the network of UNet before, and then we just find the mask during the time lapse, that is equally very fast. So there are no delays there.
Yeah, that's great. So for the MPC, do you imagine to use a stochastic model or just a deterministic model?
For the
Because
MPC,
I mean, if you are going to use MPC, like the online optimization process is going to be time consuming if it's stochastic process then.
Indeed, at the moment we are using the deterministic, otherwise the optimization would take time. You are absolutely right. There is again to say that, as I told you, anyway, we, at least for the mammalian cells, we have this 15 minutes wait to don't stress cells. But so it might be fine. Still, we didn't use the stochastic before. Again, for these exogenous systems, things were easy. So we said, okay, let's start easy. We are using a very simple linear model and is enough. Now that we are switching to endogenous processes, I can tell you that is not great. The issue, more than noise, the big issue is the fact that we fit also because of noise. If we fit the parameter of the system offline, very often when we do things online, because of noise, things are different. So another option to still keep it fast, even for the optimization, but to have something that works better, and is what we are doing at the moment, is to move to adaptive strategies, so that each time we basically take an image, we quantify the output, we apply our Kalman filter. It's something that I didn't mention, but we also use Kalman filters because we observe one output, but we have got more than one variable in the model. So we also need to estimate the other states. But if we can estimate the parameters each time we take an image, in principle, our model should be much more reliable.
Okay, thank you so much.
Thanks to you for the question, really interesting.
Okay, I also had a question about the cancer therapy. That sounds very interesting. So with your controlling the doses of the different drugs, so you think you can then like advise how, like in which proportions these drugs would have to be given, or could you also do like a time course? First it would have to be this, and when the cancer has reached a certain stage.
This will be, okay, I don't think we will have any information about the stage of cancer, mainly because we will be working with cell lines, firstly, so, you know. And the other thing is that in vivo and in clinical trials, they started to see resistance to drugs, that is what we want to avoid, but after treating patients for 1 year, and we can't do experiments that are 1 year long. Although the new device that we just built, it should allow to keep cells there for a couple of weeks, while with the actual device is like 3 days, then they die. We can do experiments that are that long. Still, to recapitulate different stages of cancer and of acquisition of resistance, what we will do is to use cell lines that have got mutations which are acquired in vivo after patients are exposed for too long to these drugs. So AstraZeneca, that is a partner in the project, they developed by CRISPR various cell lines which recapitulate these different stages of cancer. So won't be done on cancer, but on cell lines, to recapitulate that. To answer your first question, yes, in principle, we hope that combining control and possibly also optimal experimental design, we might also use that kind of approach because the parameter space is big here. We have got lots of drugs. We hopefully should be able to suggests both the dose of drug and the time of administration that basically optimizes for this phenotype you want, which can be reduced proliferation or reduced activation of signaling pathways involved in cancer, but avoiding a continuous exposure to drug. That's the idea. And at present, that is mainly done by companies using models. But again, the issue is that these models are often derived, pulling together lots of data and parameters from different data sets, which is a bit tricky. The idea would be to use the chip to have a very controlled system and the control algorithm to help us in identifying the best combination of drugs. That's the idea. And also looking forward, the idea would be to then move to organoids, which should recapitulate better the physiology of an actual organ as compared to cell lines. But we will now start with cell lines today because it's easier.
Oh, very good, cool. Are there any more other questions? Because I'd have a final one.
Yeah, if I may, can I ask one more question?
Yeah.
Yeah, I really like your talk. That's why I have so many questions. So for the multiple gene regulation part, where you show the target is repressed by the comparator, like B molecule B or something.
Yeah.
I'm wondering, so that would work if you have more target than your reference, right? Because you're repressing the target production. But what if you have less B than your reference? How are you going to boost the production then?
Okay, so you're telling me that if I don't have enough, enough basal targets, otherwise it will switch off the targets, right? Basically. Yeah, it's true. You're absolutely right. And indeed, in the first implementation of the targets we had this problem that the inhibition was too strong. So at some point, our targets were off. And the nice thing is that at least we can predict some of those things from the model that we have, the one I showed you. And another thing that we found from the model that is important is that the strength of that inhibition, absolutely right, otherwise we switch off everything. Another thing that is important is the number of cells, and that is something that in BSIM we can represent. So also how many controllers and targets you have matters. And maybe even if we would have some of the interactions that are too strong, playing with the number of cells, maybe we can adjust for those kind of things. And thirdly, something not intuitive that we found out with the simulations is that it's also really important the distance of cells. So if they're not well mixed, you lose the communication because they are too far away. So the inducer molecules can't physically reach the cells there. And that's why actually something else that we are doing at the moment is also to work with bioreactors, because it might be better actually to then move to bioreactors in such a way that for sure they are well mixed and they can interact well with each other. But you are absolutely right, Sean. That is a very important parameter for the system to work, yes.
Okay, thank you very much.
Okay, if there are no other questions, then I'd like to finish by asking you, I love this question if I can ask it, because your background is not in biology, but in maths and informatics. So when and how did you get introduced to synthetic biology?
Well, Corina, my PhD was, okay, it was within automatic engineering, but was focused on synthetic bio. And I mean, I'm not as young as you, but I think I was one of the first to have a PhD in synthetic bio. I was quite lucky in that. Although I was focusing on the modeling. So at the time we published the cell paper and I was the modeler and there was someone in the lab building the circuits in yeast. And the point is that at the end of my PhD, although I was interacting with Irene, the biologist, a lot during my PhD, I was feeling like I didn't know enough of biology. So I chose to do a postdoc that was really lab-oriented. So my postdoc in Spain with the EMBO fellowship, I was doing also the experiments for that project. There was a quite steep learning curve because I'd never been in a lab before. But honestly, I found that particularly helpful to understand more the biology by doing this. And also it helps me now in supervising also experimentalists. So it maybe slowed down a bit the publications, I have to say, because there was like 1 year of training, as you can guess. But it was really helpful. So to learn by doing, basically, yes.
And did I see you did something with iGEM as well?
Yes, yeah, I was part of an agent team the first year of my PhD, ages ago. Yes, I think it was 2007. It was really funny. And then this BSIM, this tool that I showed you today, actually went to iGEM when I wasn't in Bristol yet, but my colleagues were leading an iGEM team at the time as well. Yeah.
Very nice. It's nice to see that there's now a generation of PIs coming that actually did participate in iGEM themselves.
Yes.
Well then, if there are no other questions, thank you very much, Lucia, for the talk.
Thanks to you.
And well, thank you for joining us today. There will be another webinar next week on Friday by Jim Collins on harnessing synthetic biology to fight pathogens. We'll send out all the information in a follow-up email to you. So thank you very much for joining us today.
Thanks to you. Have a good afternoon.
You too. Bye.
Bye.